Okay, well, thank you so much for the introduction and for the invitation to join you all this morning to talk about SARS-CoV-2 and COVID-19. And also thanks to my colleague, Dr. Rajan Gupta, for arranging this session. I'm really privileged to be able to share with you some information this morning. And so, okay, let's get this working. So here's an outline of what I'd like to discuss with you today. I want to give you a brief introduction to viruses and then talk a bit about SARS-CoV-2 or SARS coronavirus 2, the virus that causes the disease, COVID-19, and then we'll talk a little bit about the disease. And then we'll talk about vaccines and therapeutics. And then I'd like to end the lecture talking a little bit about my thoughts about the future of COVID-19. And then I'd be happy to answer any questions that you may have. So let's dive right in. So first, let's just talk a little bit about what a virus is. So a viral particle, or what's known as a virion, is a very simple organism. So it has genetic information, and viruses can have DNA as their genetic information, as we do, or they can also have RNA as genetic information. And in fact, coronaviruses use RNA as their genetic information. And then that information is inserted into a protein coat that protects the DNA or the RNA from the environment. And some viruses also have a lipid envelope. So you could think of this the same way as one might the surface of the cells in our body, very, very similar. And so these are just some electron micrographs of different viruses that you may have heard of. Norovirus, which causes gastrointestinal disease, influenza virus, I'm sure you've heard of, and herpes simplex virus. And these are pretty much to scale. The smallest of these are the noroviruses. These are about 30 or 40 nanometers in size. So remember, a nanometer is 1/1 billionth of a meter. So these are very, very small, and we need what's called an electron microscope in order to take these photographs. Now, the really important thing to remember about viruses is that they can only replicate inside a living cell. So these viruses, if they're out in the environment, they are inert. They cannot do anything. They have to be inside a cell in order to be able to replicate. And so viruses do not grow and divide like cells do, but instead what they do is they turn the cell into a factory. And that factory produces the parts that allow them to assemble new viral particles or new virions. So it's a very different means of replicating. And there's a lot of debate that's been ongoing for many, many decades about whether viruses are alive or not. And it's sort of an interesting debate. Okay, so I just wanted to show you the typical life cycle of a virus, just so you can understand this in a little bit more detail. So the virus has to attach to the surface of the cell, and there are normally molecules on the surface of the cell that allow for that attachment. The virus then gets inside the cell and releases the DNA or RNA into the cell. And then that DNA or RNA will be replicated to make new copies of the viral genome. And it will also serve to produce new viral proteins. And those proteins and new genomes will come together in the assembly process to make new viruses. These are called progeny viruses. And those viruses can then be released from the cell. And different viruses get out of the cell in different ways. And we don't have to worry too much about that. The one step I want you to just pay attention to here, because we're going to talk about it a little bit later, is this attachment step, where the virus first binds to the cell before it's able to get into the cell. Now, when viruses infect their host, and here we'll be talking about humans as the host, there are two aspects of that infection that lead to disease. And so the first is direct effects of the virus. When the virus infects a cell, that's generally going to be toxic to the cell. And oftentimes the virus will kill the cell. And so that sort of damage can then lead to disease. And then also, the immune system will respond to that viral infection, and the immune system can recognize that that cell has been infected. And the immune system may also try to kill that infected cell. So the virus can kill the cell, the immune system can kill the cell, and that damage can lead to some of the symptoms. There are also other indirect effects of the immune system. So the immune response also produces molecules that cause some of the symptoms that we see during a virus infection, such as fever or body aches, the kinds of things that you've heard about or maybe even experienced if you've had COVID-19. So that's just a general introduction to viruses. So now let's jump in and talk a bit about coronaviruses. This is an electron micrograph of coronaviruses that's been artificially colored just to make it look pretty. And the name coronavirus comes from these protrusions that are sticking out of the surface of the viral particle that sometimes can look like crowns. So that's where the name corona comes from, because it looks like crowns on the surface of the virus. Okay, so prior to 2019, we knew about 5 different coronaviruses that were able to infect humans. There are 3 that cause common colds, so really don't cause any kind of severe illness. But then there are 2 that cause much more severe illness. The first was the first SARS coronavirus. So SARS stands for Severe Acute Respiratory Syndrome. So the first SARS coronavirus first came on the scene in 2003. And fortunately, we were able to control this using public health measures. So people who were infected with the virus were isolated so that they couldn't spread the virus to other people. Public health authorities did what's called contact tracing, so tried to find people who had been in contact with these individuals before they became sick. And then those individuals were placed under quarantine. So they were asked to not come in contact with other people to whom they might spread the virus. And so using these public health measures, we were able to control the SARS coronavirus outbreak, which is good because the mortality rate of SARS coronavirus was about 10%. So that's a pretty deadly virus. The second severe coronavirus is MERS coronavirus, or Middle East Respiratory Syndrome coronavirus. This outbreak began about 10 years ago in 2012, and it was also been controlled using these same public health measures. Now, the way that these viruses make their way into the human population is through a process called zoonosis. So zoonosis means the movement of a pathogen, it could be a virus or a bacterium, from an animal to humans. And so humans are always being exposed to animal viruses. Anytime that we come in close contact with an animal, if that animal is producing any sort of virus, we're going to be exposed to that virus. But the majority of the time, we are not going to become infected or get sick because there are various barriers to infection. There may be physical barriers. So remember how I told you the virus has to bind to the surface of the cell? Well, many animal viruses cannot bind to our cells. There may be genetic barriers, either in the virus or in us, that prevent this. And then also our immune system may prevent this successful zoonosis from taking place. And so we're always being exposed, but it's very, very rare that that jump from an animal to a human is successful. And then, of course, once it jumps from an animal to a human, in order for it to really cause a problem, it then has to be able to spread from that human to other humans. So there are 2 steps that really need to take place here. And so SARS coronavirus initially spread to humans from civets. So those are cats. Here's a photograph of one here. And then MERS coronavirus originally spread to humans from camels. Okay. However, I should point out that the original host, so the original source of these viruses were probably bats. And so it's thought what happened is that these were naturally bat viruses. They spread from bats to the civet or to the camel, and then from those animals into humans. So it wasn't a direct jump from the bat to the human. So now, what are the kinds of things that allow for viruses to emerge, or in some cases reemerge from nature into the human population? I think this is something to keep in mind because as we're going to talk about later, this could happen again in the future. So first of all, there's the spread of the vectors. And by the vectors, I mean the animals that carry these viruses. So climate change is affecting where animals live, where their natural habitat is going to be. And so that's then also going to potentially lead to more human-animal contact. And the other things that contribute to this are urbanization. So as there's more sort of sprawl of the population and economic development, as humans sort of infringe on the animals' natural habitats, then there's going to be more of these types of contacts. And so it's been estimated that there are well over 320,000 mammalian viruses out there. Many of them are in bats. It's a really interesting area of research trying to understand why bats seem to carry many, many viruses. But the question that we don't know the answer to is how many of these 320,000 viruses have the potential to spill over into humans, because we don't fully understand all of the properties of a virus that allow for that jump to occur successfully. Okay, so let's now talk about SARS coronavirus 2, the second SARS coronavirus. So it was first detected in late 2019 in China. You probably all know that the original source or the original site in which the virus arose is in Wuhan, China. It's highly contagious. It has a high mortality rate. So in 2020, before we had vaccines or any sorts of therapeutics, the mortality rate was anywhere between 3 and 7%. That's dropped now. In general, the mortality rate now is more about 1% in the total population, mainly because many of us have been vaccinated. And in March 2020, so just a few months later, the WHO, the World Health Organization, declared a pandemic. And so what's a pandemic? So there are two terms here that are important to know. An epidemic is when there's an outbreak of infection in a certain geographical area. So for example, right now in the United States, we are experiencing what we call the opioid epidemic, where there's been a lot of misuse of opioids leading to many, many deaths, okay? And then for a pandemic to be named, that outbreak has to spread over multiple continents. And again, so this spread from China in the fall of 2019, late fall, throughout the world by March 2020, very, very rapid spread. And of course, this is facilitated by things like air travel and the like. And so here's the current data. As of this week, over 500 million people have been infected. And this is probably an underestimate of the number of people who have been infected because of limitations in terms of testing. Also, in terms of many people have infections that are not symptomatic. And so we aren't able to detect those. And there have been over 6 million confirmed deaths. And this is a map that I pulled from Johns Hopkins University. They have a very nice website that tracks the pandemic, and it just shows sort of the distribution of infections globally. And the red circles here, the size of the circle is proportional to the number of cases in those parts of the world. So here's the US over here, and you can see India over here. Okay, so what makes SARS-CoV-2 so severe? Remember, I told you there's some coronaviruses, right, that just cause common colds. you get sick for a couple of days and then you feel better and everything's fine. So why is this particular virus so severe? So there are a few things that contribute to that. The first is that this virus has the ability to infect our lower respiratory tract. So the cellular receptor, so the molecule on the surface of the cell that the virus uses to get into the cell, is made by cells in the lung. So this virus has the ability to infect cells in the lung very well, whereas the common cold coronaviruses can't do that. So that's one problem. The second problem is that SARS-CoV-2 can be spread by asymptomatic individuals. So why is this important? It's important because it makes public health measures difficult. Remember how I told you about the first SARS coronavirus and MERS coronavirus, that public health officials were able to do isolation, contact tracing, quarantine to limit the spread. But all of that was dependent on people being symptomatic. So the first SARS coronavirus couldn't spread until people had symptoms. This virus can spread before people get symptoms, or even in individuals who never have symptoms, but are still producing the virus. And that's what made public health control of this virus so very, very difficult. And then finally, because this was a new virus that jumped into the human population, nobody in the human population had any existing immunity to the virus. And so that then allows for the virus to replicate to a very high level before the immune system can do its job and try to interfere with the viral infection. And so these three parameters here are what make SARS-CoV-2 infection so severe. So I need to talk a little bit about variants. We've all heard about variants, and I think this is a really interesting topic, and it's something that we will need to continue to pay attention to over the coming months and years. So how do variants arise? When viruses are copying their genomes to make new viruses, the enzymes that do that make a lot of mistakes. And so they make many mutations and introduce many mutations into the new viruses that are being produced. And these mutations can alter a number of different things. So for example, they can cause the virus to be able to replicate better. If the virus replicates better, that means that each time the virus infects a cell, that cell is going to be able to make more viruses than the original virus before it had the mutation. Some of these mutations affect the ability of the virus to bind to the cell and get into the cell. So if the virus can do this better, it's going to be more efficient at infecting new people. The stability of the viral particle itself. So the more stable the virus is when it gets out into the environment, then the more chance that someone else may be exposed to that virus. And so this then affects the ability of the virus to transmit from one person to another. It could affect the virulence or how severe that infection is. And it can also affect the recognition by the immune system. So these variants may not be recognized as well by the immune system. And so let's talk a little bit about, how this is sort of played out over the past couple of years. And so the SARS-CoV-2 variants that we've seen, and you've heard about them, right? Alpha, Delta, Omicron, all of them have shown increased transmissibility, right? So their ability to spread from one person to another has been increased. And I want to introduce a concept to you that is used by epidemiologists, so individuals who study epidemics, and it's called the reproduction number, or R naught. So the way to think about the reproduction number is, how many people is an infected person likely to spread the virus to? Okay, so for the original Wuhan virus, it was about 3. For Delta, it was about 5. And for Omicron, we don't really even know the real number right now because those data are still being collected. It's probably somewhere between 7 to 10. And so let me illustrate this for you. So here's the original virus. And let's say there's someone here who's been infected. And that person is going to spread it on average to 3 other people. And each of those people is going to spread it to 3 more people. So by the second round of spread, it's gone from 1 person to 9 people. Okay. Now let's look at Delta. So here's the original person who's infected. That person's going to spread it to 5 other people because the R naught is 5. And each one of them is going to spread it to 5 more people. So now in the second round, you've got 25 infected people. So you can see how this grows exponentially, right? So even though these 2 numbers are not that far apart, they're not even double each other, the actual effect of here is a lot more dramatic. So you go from 1 to 9 versus 1 to 25 very quickly. And that's why Omicron has spread so rapidly throughout the world. Now, these variants can also sometimes evade the immune system. And so you've probably heard that the Omicron variant is not as well recognized by the current vaccines. Okay, and that's because the mutations in Omicron not only affect its ability to spread, but also affect its ability to be recognized by the immune system in individuals who have been vaccinated, because the vaccines are all using the original strain of the virus. Okay, so let's talk a little bit more about these vaccines. And I just want to introduce you to the different vaccine technologies that are being used throughout the world. I can tell you that at the beginning of the pandemic, if you had asked me how long it would take to develop a COVID-19 vaccine, I would have said at least 2 years. And remarkably, we had vaccines within 9 months. So it was really an amazing feat of biomedical science in order to be able to develop these vaccines so rapidly. And I think it's just a measure of the global cooperation that we had in studying the virus, understanding how it replicates, understanding how it causes disease, and then trying to develop vaccines. So there are 2 key parts of what's called the adaptive immune system, so the immune response against a pathogen that we'd like to be able to stimulate. The first are antibodies, and you've all heard about antibodies. And then there's what's called cell-mediated immunity. So this is the part of the immune response that I talked about early on that can recognize an infected cell and kill that infected cell. So the major goal of COVID-19 vaccines, and of many vaccines, is to generate what are called neutralizing antibodies. So these are antibodies that interfere with the ability of the virus to bind to the receptor on the surface of the cell. And so therefore, they block the ability of the virus to infect the cell. And so I'm just going to show you a little cartoon of how that works, right? So here's SARS-CoV-2, and on the surface, there's this protein called the spike protein that you've probably heard about. And it's this spike protein that allows the virus to bind to the molecule on the cell surface. And then the virus will, from there, be able to enter into the cell. And so a neutralizing antibody will bind to the spike protein and prevent it from being able to interact with that protein on the surface of the cell. And if the virus can't interact with the cell, it can't get inside the cell, and so you get no infection. And so neutralizing antibodies are really sort of the best type of immune response you could get from a vaccine because it prevents the virus from infecting the cell in the first place. Now, we talked about how some variants may be able to evade recognition by antibodies, but that's okay because, again, even once they do get into the cell, then that cell-mediated immune response is going to come in and kill that infected cell. And that's why vaccinated individuals get much less severe disease. They're much less likely to be hospitalized much less likely to die because that adaptive immune response can still come in and do its job. And so here are the major types of COVID-19 vaccines that are being used. So there's what are called mRNA or messenger RNA vaccines, where it's a messenger RNA that's complexed with nanoparticles. So these are really, really tiny particles that allow that mRNA to get into the cell when they're injected into your muscle. And that mRNA gets translated by the cell to produce the spike protein. And then the immune response recognizes that spike protein and builds up immunity against the spike protein. There are viral vectors. So what people have done is to engineer other viruses that don't cause disease to produce the spike protein. So you could think about it as using another virus as a vehicle to introduce the spike protein into cells so the immune response can recognize it. There are what are called subunit vaccines. So this is where the spike protein is produced in a laboratory to very, very high levels. That protein can be purified and then injected directly into individuals to induce an immune response. And then there are inactivated viruses. So what's done here is to take authentic SARS-CoV-2 virus and treat it in such a way to basically kill it, so it's not infectious. And this can be done chemically, it can be done using heat or other mechanisms so that the virus will be recognized by the immune system, but it can't cause any disease. And so I just wanted to bring you up to date on the progress with respect to getting the world vaccinated against COVID-19. To date, over 11.5 billion doses have been administered. That sounds like a really large number, but what we have to keep in mind is that the population of Earth is 7.5 billion people. Many of these vaccines require 2 or sometimes 3 doses in order to be fully effective. And so right now, about 4.6 billion people have been fully vaccinated. So about 59% of the world's population. And I just pulled these numbers a couple of days ago, again, from the Johns Hopkins website that I told you about. In India, there's been almost 2 billion doses administered, and about 61% of your population is fully vaccinated. So I think India is doing a really good job here in terms of getting individuals vaccinated. Okay, let's talk for a minute about therapeutics. So the number of therapeutics that we have available to treat COVID-19 are limited right now, but many more are under investigation. One of them is a drug that inhibits replication of the virus. It's called remdesivir. This is a bit limited in terms of its utility, though, because it has to be given intravenously. So this can't be taken at home as a pill. It has to be administered in a clinic or a hospital. So the delivery of this drug is a little more difficult to accomplish. But there are 2 medications that can be given in a pill form that you can just swallow. And they inhibit other viral proteins. These are the names here. You don't have to worry about what the names are. But the key thing about all of these therapeutics is that they work best if they're given to people as soon as they become symptomatic. So if you wait too long to give these drugs, then they're really not effective. And the reason for that is that they're going to work best when there's not a lot of virus in the individual, right? So then they have a better chance of knocking it down. And then you've probably also heard about monoclonal antibody treatments. So monoclonal antibodies are antibodies that can be produced in the laboratory. So in a way, you could think of these as sort of a substitute immune system. So rather than vaccinating someone to induce immunity, you can just inject antibodies into a person that recognize the spike protein and therefore will prevent any virus in that individual from infecting the cells in that individual. Monoclonal antibody therapy is quite expensive, though, and so it's not quite as available throughout the world, as are some of these other therapeutics. Okay, so I want to finish up just talking a little bit about what I see as the future of COVID-19 and just present you with a few questions to think about. So the first is, will enough people ever become immune? So how will people become immune? They're going to become immune either by having been infected with the virus or through vaccination, right? And of course, some of the things that will affect that are vaccine hesitance. So we know that there are many people who are skeptical of the COVID-19 vaccines for various reasons. Here in the US, sometimes those reasons are political, sometimes they're not. And then there's also the issue of the distribution and cost of vaccines. So how do we deliver vaccines efficiently to third world countries that really cannot bear the cost? And there it's really going to be up to the developed world to step in and make these vaccines available to places in the world where they're sort of not affordable. The second question is, will variants continue to be problematic? So my view about this is the best thing we could do is to try to prevent variants from arising in the first place. So how do we do that? We do that by limiting how much the virus can replicate. So the less the virus replicates, the fewer mutations that are going to arise, and the less chance of getting a variant that's going to cause a problem. And of course, the way that we lower viral replication is through immunity. So these two are sort of related. Now, one thing to keep in mind here is that there are animal reservoirs of the virus. So what does this mean? This means that there are animals that can be infected with SARS-CoV-2. Here in Michigan, we have deer population, and the deer population has been infected with SARS-CoV-2. And so even if we can really knock down infections in humans, there's always the possibility that the virus is in one of these animal reservoirs and then potentially can mutate there, leading to a new variant that can then jump back into humans. And there's some evidence that the Omicron variant may have actually done this. In other words, what may have happened is that humans were infected, a human infected some animal, it spread through those animals and then came back into the human population as Omicron. So that's a problem that we need to consider. Will SARS-CoV-2 ever be eliminated, or will it become pandemic? I do not think it's going to be eliminated just because of some of these issues that we're dealing with, where we're not going to be able to get everyone vaccinated. There's going to be this issue where the virus can potentially jump back and forth between animals and humans. And so I think it will become endemic. So endemic means it's basically going to become another virus that we just live with, but that we have the ability to prevent from causing severe disease. So it'll become like influenza or colds or other viruses that mankind has lived with for many, many, many years. And then the final question to think about is, will another coronavirus make the jump to humans? So I told you, just in the 21st century, we've had 3 new coronaviruses jump into the human population. And so the question is, will another coronavirus do this at some point in the future? And if so, will we be better prepared than we were for COVID-19? And of course, this is going to require a lot of political will in order to be able to deal with this potential eventuality in the future. And so that is my introduction to SARS-CoV-2 and COVID-19. Thank you very much for your attention.